Good afternoon, everyone, and welcome to today's session hosted by the Latin America Society for Immunodeficiency. We are the Juniors Committee of the Society. We are very pleased to have with you. We have to have you with us for what promises to be a fascinating and insightful talk. It's a privilege to introduce our guest speaker today, Dr. Jorge Gonzalez. But first, we are going to have some messages to take home about our society. If we have the slides, please, Alfonso. Well, first of all, remember that we have this last meeting taking place here in Buenos Aires in October, so please say the date and came to join us. And also, we want to tell you that the call for abstracts has been extended to the last day of June, so you have time to submit your work, and we will hope to see you there.
Well, good afternoon, and also we want to invite you to become readers and also contributors to this newly Journal of Human Immunity. As you know, it's officially recognized as the scientific journal of the different immunology societies, CIS from USA, also ESIT, and of course LACIT. This journal serves as a platform for advancing the field in immunodeficiency in human immunology through high quality research. So it's an important tool that we have to take into account. The next slide, thank you. And also, we want to invite you to become members of our society, LACIT, and to have an active participate in our academic and educational activities through year this, especially this year that we have the LACIT meeting in Buenos Aires. So it's very important, your engagement is essential to foster the vibrant and collaborative immunology community.
So, as I said before, our guest speaker today is Dr. Jorge Gonzalez. He's a dedicated researcher who work lies at the intersection of immunology and rheumatology. It's very interesting for us. His scientific contributions continue to shed light on complex mechanisms involved in autoimmunity disease, and his perspective brings valuable insight into both clinical practice and experimental research. Rafa, if you want to continue.
I'm sorry. Dr. Gonzalez completed his medical studies at the Faculty of Nuevo León in Mexico. He later pursued his master's degree and a PhD in immunology at the University of Hamburg in Germany. And he's currently based in Seattle, where he conducts laboratory research at the University of Washington within the Department of Rheumatology. So his work is focused on understanding the role of mitochondria in various autoimmune diseases.
So we warmly invite Dr. Gonzalez to join to this session. It's a pleasure and it's an honor, a true honor to have Dr. Gonzalez with us in your region also, Latin America. So please be welcome, Jorge, and thank you for your acceptance to our meeting.
Thank you everyone for the invitation. It's really an honor for me to be invited to this really big meeting because it's big. It even sounds here in North America. So a lot of people knows about LACID. So for me, it's a big honor to be part of this. And I think this is the moment where I need to start sharing my slides, isn't it? So I will start sharing it. Just let me know if you can see my presentation.
Yes.
Is it good? Okay, perfect. So basically what we will be speaking about in the next few minutes is just like a kind of a work in progress. What we are trying to do with certain kind of diseases, in this specific case, myopathies like dermatomyositis and juvenile dermatomyositis. So we work in conjunction with really a lot of different people from different specialties, especially immunology, especially rheumatology. So I will share some of the things we have been doing, some of the things we have been thinking on doing, and some of the results, some of the collaborations also we have been doing, like some of them are pretty amazing also. So okay, so first of all, we will need to, I'm pretty sure most of you know about what is dermatomyositis, or which kind of diseases are this, but just as a friendly reminder, so what's the difference, or what they have in common in this case, juvenile dermatomyositis and dermatomyositis? So one of them is basically during childhood, the other one is in adulthood. So, but basically what they have in common is that they have a chronic curse. So this is like a chronic disease. It's not something that is acute and it will be solved the next day and that's it. So it's chronic. Second, they have muscle involvement. Of course, as the name says, derma, toe. So it includes also other kind of tissues and in the human body, like skin, for example. But our focus is mainly muscle, skeletal muscle. So it has muscle involvement. So it creates weakness in the patients, and this is associated with a lot of complications and comorbidities. So some of the complications or comorbidities in these patients are actually pretty interesting because these patients may develop, not everyone, but 20% of the patients with dermatomyositis and 40% of the patients with juvenile dermatomyositis may develop something called calcinosis. So basically, this is the accumulation of calcium, specifically hydroxyapatite crystals of calcium in soft tissue. So this soft tissue, of course, includes the muscle. So some of the pictures of the patients, they may be looking like this. Some may be looking pretty much dramatic, like this one. Some other ones, you will be able to see them in the x-rays, like all of these deposits here, or even you can even touch them like in a simple clinical examination of the patients. So this is kind of important because these calcinosis may get infected because they will try to escape the body basically, and they can get infected and that have like consequences and complications. So basically these patients, besides being affected like in skeletal muscle, in skin, having the complications of calcinosis, they have many other things involving the disease. So the quality of life of the patients will be deeply impacted. So they will have a very low quality of life, both children, which will become more dependent to adults, and adults also, which will become more dependent to other adults. So simple tasks in life will be basically 100% dependent to other people. Also interesting, this is why partially why we are here also in Seattle doing our research, is that both diseases are classified, or many people classify them as mitochondrial diseases. This means that they have something in their mitochondria that is not working well. In other words, they have dysfunctional mitochondria. What is happening exactly? We don't know. So we're trying to figure it out. But we have seen in different patients that certain types of mitochondrial biomarkers are elevated in circulation in plasma in these patients. So we have been measuring something called formulated peptides and also GDF15. So formulated peptides are a specific kind of peptide that can come basically from two sources, or it comes from mitochondria, or it comes from the microbiome. And our hypotheses and our studies are suggesting that it comes basically from the mitochondria. So why this kind of link? If we remember many billions of years ago, mitochondria, they were a bacteria, they create a symbiosis with another kind of cell, and they create like a symbiote. So basically, mitochondria and bacteria have many things in common. And one of those things in common are the formulated peptides. So these patients have really high levels of this protein in circulation. And together with this, there's something else called GDF15, which is the differentiation growth factor 15. This, contrary to the formulated peptide, which can be pro-inflammatory, this is an anti-inflammatory cytokine. So many patients have this one also elevated, probably trying to contrast or trying to kind of balance the inflammatory response in patients with high mitochondria or extracellular mitochondria content. So there is kind of a very intriguing aspect with GDF15 as an actual discussion. So many people don't know actually where it's coming from. So it can come from different tissues like muscle, skeletal muscle, but some studies suggest that it also may come from mitochondria itself. So, and that's something we're also trying to figure it out, like where is it specifically coming, and if it has any other function besides just being an anti-inflammatory cytokine. So together with this, we have found also some interesting data that we have elevated levels of mitochondria biomarkers and anti-mitochondria antibodies, which are associated with calcinosis. So basically, the patients that were having higher amount of anti-mitochondria antibodies were having or were clinically having a worse calcinosis. So basically, more severe disease with more complications, more comorbidities, and more inpatient time inside the hospital, basically. So that was something also interesting we found. So something we must remember about the mitochondria is that they have besides being the powerhouse of the cell, that's basically what we all know mitochondria for, the powerhouse of the cell. They have many other functions, many other functions. They regulate apoptosis, they regulate like calcium homeostasis, but they also regulate somehow the interferon response. They have also their own antiviral response. So the interferon production in mitochondria myopathies or during a mitochondrial dysfunction maybe also kind of be affected. So we were thinking, okay, these patients have calcinosis, mitochondria are dysfunctional, mitochondria regulate calciums, so probably this calcinosis is affecting also mitochondria, which is releasing mitochondrial DNA and many other proteins that per se will be immunogenic and will create an immune response. And since the interferon response is mainly driven by this pathway, which is the C-gas sting, we were thinking, okay, what will happen if we basically block the C-gas sting pathway in these kind of patients and samples, and maybe they will have like less mitochondria antibodies or less kind of clinical severity of the disease. So, and we actually saw something also interesting here. So basically, I don't know, you can see this is kind of a blood vessel. This is a muscle, skeletal muscle. And we were seeing like 3 extracellular mitochondria. And not just that, these mitochondria were calcified. So something were happening or is happening in the patients that mitochondria were being released, which is a phenomenon that usually happens. I mean, we all have circulating mitochondria in our body, but the problem here is that these mitochondria were calcified, and that's not normal. So, or these mitochondria may be dysfunctional and may be releasing some proteins, some mitochondrial DNA, and that will be causing an immune response. So we're able to find like these mitochondria, and we're seeing like, okay, maybe this is triggering a very strong immune response in these patients. So we started like some of my colleagues also started like inhibiting certain pathways of the interferon production of the C-gas sting. So like STAT3, for example. So we can see that at the moment of adding certain STAT3 inhibitors, uh, basically the osteo image, the median fluorescence intensity of basically the calcium was kind of even like almost half of the one compared also with the only one of calcium phosphate and interferon. So we can see here like just the media, so nothing happens in the muscle. Interferon response, I mean just interferon, nothing happens. When we combine calcium and interferon, boom, we have a big response. And when we combine calcium, interferon plus the STAT3 inhibitor, boom, there is a drastically kind of lowering down the levels of this osteo image. And something similar happened to the interferon-driven genes like this one, which was basically also damped or lowered by a STING inhibitor, which is this one, H151. So, and the same thing happened at the moment of inhibiting like mitochondrial ROS production through something called mitotempo. This, what it causes is that it will go inside the mitochondria, it will stop the reactive oxygen species, which may be very toxic, and that will be in theory causing less inflammatory response. So basically, the inhibition of the interferon production in this experimental model showed us that it was causing less calcification, at least in this in vitro model. So that was something very interesting. So you may be kind of asking, okay, so far you have spoken about mitochondria. They're outside the cell. They may be having calcifications and interferons production, maybe also causing this calcification. So that's kind of the idea. So why is this kind of important? Because these kind of diseases, they DM and JDM, they have a lot of, the unknown kind of unanswered questions. So these will lead us basically to discover new biomarkers of the disease, which may be related to mitochondria specifically, new biomarkers which will be related also to calcification or even calcification prevention, new autoantigens, which will be directed to specific antigens that may be also coming from mitochondria. And these may have like clinical significance. And this will also probably help us to try to regulate the type 1 interferon production or response in the mitochondria kind of metabolomics. So it has like deep impact in these specific diseases. But this being said, this can be also translated into or translocated into other kinds of diseases because there are like many mitochondria diseases. These are just a couple of them, but there are many mitochondria diseases. And if this is a trigger here, probably maybe they have something in common with other kind of diseases. So this is kind of important. So the main goal basically of our lab and of our team is to investigate mechanisms of mitochondria-mediated inflammation in autoimmunity. This includes JDM, DM, and we're working with also IBM, which is in the line of rheumatology and neurology. It's kind of a more degenerative disease or inflammatory disease because you can see some CD8 cells over there. So that's kind of a blurred line between rheumatology. It's kind of very interesting disease. We can speak about that later if you have any questions. But basically, it's for identify biomarkers and some therapeutic targets or new therapeutic approaches into these diseases for improving the patient's outcomes. So we have different objectives here. So first of all, maybe you were asking this earlier also, but okay, why we have extracellular mitochondria? So why they are outside the cell? In theory, they're the powerhouse of the cell. So what's the function outside the cell? So that's unknown. So nobody knows that. So that's one of our objectives to try to understand what is happening here. If they have that, they must have any other hidden function for us. So we're trying to determine that specifically in this context of JDM and DM, of course, and to investigate the role of interferon signaling in mitochondrial function and the calcinosis. So since we have shown that the interferon signaling is important for calcinosis in these patients, we want to know more about this. We want to know if there may be some specific therapeutic target that we can be like aiming for to try to regulate or prevent calcinosis in these patients. Because one is developed, if they're not treated correctly, they can be removed surgically, but they have a lot of complications. So the rationale behind these objectives are pretty simple. So in these kind of diseases, there's a lack of reliable biomarkers. So we have like creatine kinase, CK, which is commonly used for muscle involvement in many other kind of diseases, but it's not very specific. So we're looking for something more specific. So there's also mitochondrial involvement, which is very interesting. Want to uncover like what else are they doing over here besides providing energy, regulating calcium, regulating apoptosis, or even immune response. So we want to know what's happening with the formulated peptides, which are the FMET, because something very interesting is that formulated peptides, they have a specific receptor in some cells, specifically neutrophils. So neutrophils have something called FPR1. So formulated peptide receptor 1. So this receptor is made specifically for these kind of peptides. There's a reason for that, and we want to know why. So if we have a lot of free mitochondria, if we have a lot of free formulated peptides, maybe neutrophils are playing like a very important role in these kind of diseases and many other autoimmune diseases. But maybe we're just kind of overseeing them because they're like the most common cells we have, the white blood, white cells. And also we want to know or study more the neutrophil activation, if they can predict the treatment response, probably to JAK-STAT inhibitors, like in this case, which has not been published yet, with baricitinib. That basically the neutrophil response was predicting basically the treatment in these patients in another kind of disease, but we want to switch it to the DM and to JDM. So maybe neutrophils that can respond better to certain kind of drugs will predict the better or the worst outcome of the patients. So this is our main hypothesis. We have been thinking for this only first objective. So these biomarkers, they will be associated with key symptoms of DM and JDM. They will be associated with the severity of the disease and with the calcinosis. So, and we will also kind of, unveil how mitochondria will be influencing the inflammation, how they will be activating neutrophils, mainly to formulated peptide receptor 1, because that's the main receptor neutrophil has for mitochondria, or that's what we know so far. So we have like we are approaching this like in different ways. So for the biomarker part, we have collected like many patients, or some of them were given to us, the samples from different institutes. I have them like in the last slide, also as a thank you. So we collected like plasma samples. We have been measuring different kinds of peptides, in this case, formulated peptides, MTND6, which is a subtype of formulated peptide. We have been measuring GDF15, typical ELISA, sandwich ELISA. It's not a lot of science over there, but we are correlating them and associating these kinds of levels with comorbidities, with muscle test scores, with other kind of biomarkers that are already available, like CK. So we want to know if these ones are very specific biomarkers for these kind of diseases. And not just that, we're working also on a longitudinal cohort. So like 1 month, 3 months, 6 months, and this will be helpful at the moment of trying, to monitor the treatment response in these kind of patients. So that's kind of the first step. So for the inflammatory capacity of the formulated peptides, so we're doing a very interesting experiment, actually. So we isolate neutrophils, which is a pretty easy thing to do. So, and we are basically culturing them with plasma of healthy controls and plasma of DM or JDM. So, and then we will be measuring through flow cytometry, like activation markers of these neutrophils. So we will combine the biomarkers, we will combine basically the activation markers of the neutrophils, and that will give us an idea if these neutrophils are actually more active in these kind of diseases. If the plasma from DM or JDM have something that are activating these neutrophils. So this doesn't actually answer the causality, of why neutrophils are activating themselves during DM or JDM plasma exposure. So for answering this question, since we know neutrophils have a specific receptor for formulated peptides, what we're doing is we're inhibiting this specific receptor with cyclosporin H. So we inhibit that receptor. If nothing happens, even if they're exposed to the JDM and DM plasma, okay, that means that the main driver, the inflammatory response of the neutrophil is mainly driven through this receptor, and the receptor recognizes formulated peptide, basically. So that will explain causality in these experiments. So I will pause a bit here just in case someone has questions, unless the program states that the questions are at the end. Otherwise, I will just continue. I think the questions are at the end, isn't it?
Thank you, Jorge. I think that we can move on in questions at the end of the presentation.
Perfect. That's perfect. So We will keep going with the second objective, which also gets very interesting. So this one is to investigate the role of the interference signaling on the mitochondrial function and also the calcinosis. So the rationale behind this is also pretty simple. So type 1 interferons, well known, plays like a very crucial role in idiopathic inflammatory myopathies. So they are very strongly associated with severity of disease. So there's a direct impact of JAK-STAT modulation downstream of interference signaling on mitochondrial behavior. So this is kind of something we have been seeing, but it remains like mainly underexplored. So we need to go deeper into this one. So we want to feel like get the gap that exists by exploring the JAK-STAT inhibitors and mitochondrial function to see if there's some kind of relation to the inflammatory response, to the calcinosis or other comorbidities, and something I'm very enthusiastic in doing, which is the potential of mitochondria transplantation as a probably therapeutic strategy. So this is basically the rationale behind this objective. So our hypothesis, basically, it was said during the rationale. So there's a dysregulation of JAK-STAT signaling in these kind of patients that is induced by interference type 1 interference. This will precipitate mitochondrial dysfunction in skeletal muscle. This will be causing the accumulation of reactive oxygen species from mitochondria that will generate calcification, and this will be creating like an inflammatory response, like an ever-ending story. So it's basically like a cycle. So our idea is that these JAK-STAT inhibitors can mitigate these pathogenic consequences, and the mitochondrial transplantation could rectify cellular dysfunction or cellular metabolic problems, attenuating these effects basically. So we have two different approaches here. So we have the approach which is mainly driven by interferon 1's response, and we have something called the hybrid model. We'll go into each one of them very fast. So this is kind of pretty simple. We have certain cell line, in this case Arch30s, which is an immortalized cell line. We're also working to not use, or going forward, to not use cancer cells, because at the end of the day, these are cancer cells, and we want to use primary cells, primary culture, or any other cell culture that is not involved with cancer, because cancer has a lot of things that we don't know. So, but it's a very first step to do, for an in vitro study. So we basically add them a specific amount of interferons, and we want to see the prophylactic effect of the JAK inhibitors and the basically therapeutic effect of JAK inhibitors. So we want to see like both sides, how will it work better, basically. Will it be working better like as a prophylactic, or will it be working better like as a therapeutic? So we want to explore like both kind of sides in this kind of experimental model. So of course we will add patient-derived plasma from DM and JDM, and we will use also like STAT3 inhibitors like this one, just to try like different aspects of the whole kind of signaling of the JAK-STAT signaling. And we will measure this like in different ways, mitosox red, also calcifications, the seahorse, very important for the metabolomics part of the cells, mass spectrometry to try to identify new proteins or particles, and many other pathways that are also related to the interferon inflammatory response. And of course, we will do, or what we are doing is the immunohistochemistry for in the muscle biopsy, basically, of these patients. That will be also like something very important to do, because the in vitro model, that's in vitro, that's cancer cells, but we have the actual biopsy of the patients that will provide us also a lot of information. And the hybrid model, which is kind of very interesting. So a hybrid model is basically a model where you have like normal cells. They have like, in this case, normal Rh30s, cancer, but at the end of the day, normal for the things we want to use it for. So they have mitochondria inside. A lot of mitochondria, because Rh30 comes from a rhabdomyosarcoma, so it's muscle at the end of the day. So there are actually two models we have been using here. liver cells, which have a lot of mitochondria due to the high metabolic demand, and skeletal muscle cells or muscle cells like this one, which also have a lot of mitochondria through because of the metabolic demand. So what we are doing is basically taking those cells. We add a toxic agent. In this case, can be ethidium bromide, or there's another one called rhodamine 6G, and this will specifically affect mitochondria and will deplete mitochondria. So rhodamine 6G and ethidium bromide works differently, but the main goal is the same. Cells will be there and they will not have mitochondria. So how will they survive? You may be asking yourselves. So we basically provide them with additional food, additional media, additional kind of resources for them to keep them alive. Since they don't have mitochondria, we're using other kind of cycle in the biology for, in this case, using uridine and pyruvate for making things easier for the cells to generate energy and keep them alive. And this works perfectly. Actually, I didn't mention this. Sorry, I will go in the back one. So this is kind of a comparison of mitochondrial DNA in patients in Aristetti cells with just normal media and the cells treated with ethidium bromide. So basically, they don't have mitochondrial DNA. There's nothing there. The mitochondria itself, the structure can still be there, but it doesn't have mitochondrial DNA. And if there is no mitochondrial DNA, basically the mitochondria is dysfunctional. The same with rhodamine 6G. So the cell, the same structure of the cell, the mitochondria is there, but mitochondrial DNA cannot be there, it's depleted, so they are basically dysfunctional. So this is another example after basically 2 weeks. We were trying to see if actually the cells without mitochondria were recovering by themselves, because maybe among all the flask, maybe there's one cell that didn't get like affected, too much affected by the toxic agent. And, mitochondria, they have their own division cycle different from the cells. So maybe this cell after some weeks or after some days, we'll try to replenish or try to provide more mitochondria to the neighbor cells, basically. And we wanted to know, we were having like a window, a time window before this happened. So, and we saw that 2 weeks were basically enough for the cells, even in the recovery media, for not to recover. So this told us, okay, cells without mitochondria in the recovery media, they're pretty much stable. They are not recovering. They're not getting their mitochondria back. So basically 100% of the cells were affected by this method. And now here comes the mitochondrial transfer. So what we're trying to do here is like, there are many ways of doing the transfer. So we're just approaching several of them. So we have normal Aristetti cells that were stimulated with interferon or not stimulated with interferon. And then we isolate mitochondria and then we transfer them just simply by co-culturing them to the cells that don't have mitochondria. So we are using the co-culture method. Basically, you just add the mitochondria, certain ratios, certain concentrations, of course, like cooking literally, or we use platelets through PRP. Why platelets? Because platelets have a lot of mitochondria. And very interestingly, when platelets gets activated, they release a lot of things, and among them, mitochondria. So we were thinking, what if we isolate platelets or we use the PRP, we stimulate the platelets to get them activated, and they will be releasing a lot of stuff. But among those stuff will be also causing the release of mitochondria. And maybe the cells in need that don't have the mitochondria will be taking them. So that's basically the two methods we were kind of we are mainly using. Each one of them has their own variants. So the co-culture method, you can also make like a centrifuge, for gravity, for literally like pushing mitochondria inside the cell. The platelets, also you can use different concentrations of platelets, different activators of platelets. So they have their own variants. And of course, these are just two methods. There are like many different methods that can or probably work, but these are the ones we're trying to use so far. And after doing the transplant, Okay, we will measure like again many different kind of parameters of in these cells just to try to see if the cells are actually recovering. So we want to know if okay cells are recovering after the transplantation. So that may be like a good thing, and that would be like a very promising like therapy. You know, there's some papers out there that are actually now they're trying mitochondrial transplantation. There's some caveats in those papers, so we're also trying to fill those caveats and considering those for our model. And we're working with other companies or agencies in this case also to try to make this as easy as possible. You know, easy science is always better. The less complicated it is, the better. So basically, after in this hybrid model, so we want also to add the plasma or mitochondria from healthy controls and from dermatomyositis patients, just to try to see which kind of effect they have in these kind of cells. So we want to know if the plasma of the DM patients or JDM patients has something that is causing the mitochondrial dysfunction, and we will also see it here when we compare it to the healthy control plasma, and of course measure like a lot of different variables just to see if this is kind of creating a different effect or not. So this is one of the examples we have been doing. So we have like normal cells, the cells that were depleted with the titanium bromide mitochondria, so basically not showing any kind of mitochondrial DNA, the cells that have the recovery media, and then here we started adding the PRP. In this case, I think it was like 10% of PRP, just the PRP itself, where it showed like after 3 weeks that the mitochondrial DNA was actually going up. And when we combine the PRP plus ADP, ADP is an activator, platelets activator, this one even a bit higher. So we made this like in different weeks, because if of course if you add it and you measure it immediately, there's a probability the mitochondria might be stuck, you know, outside the cells, and when you isolate the DNA, you can be getting those kind of DNA from the stock or the stick-like mitochondria. So after washing and after different weeks, this basically shows us like, okay, there's somehow kind of recovering. And this is another example where we use even higher concentrations of PRP and different kind of transplant methods. We were using also PBS as a first step for the transplantation, and I will show you later why we're using PBS in one of the last slides, because mitochondria we found are very picky with the extracellular environment. But basically, the most important part here is that, yeah, PRP seems to be working and activated platelets seem to be like recovering the cells, you know, from that were having depleted mitochondria. So mitochondria are like very picky in the extracellular environment, and that's why we were using also PBS in the very beginning. So media, when you're using culture media or basically media for culturing cells, they have a lot of macromolecules like lipids. They have also like a lot of minerals. Some of them have a lot of calcium, a lot of phosphates. So mitochondria viability in extracellular environments it's very tricky. So high calcium concentrations or even phosphate concentrations even inside our own body in the fluids or in a cell culture may impair the function. So we were trying, you know, a transplant like in 2 steps, like maybe first step, just PBS, the transplant, half an hour, short time, and then switch into the media just to give enough time to the mitochondria to get absorbed basically by the cell. Also, ATP production. So if the mitochondria gets inside the cell, that doesn't mean it's working. So that's why we are measuring like different things like the seahorse, you know, using seahorse to actually do a metabolomics or do a metabolic activity of the cell to see if they're actually producing ATP. So mitochondria must remain bioenergetically active post-transplant. It doesn't show us anything, just a picture inside of the mitochondria inside the cell. We need to know if it's actually active. So we need to know also if it's active and if it's inside the cell, that the cell will not destroy it. So because some of the times we have seen that, yeah, they're inside like a vesicle, like an endosome, and it will be destroyed basically. So there's a lot of things. And also, if this kind of works, we need to, if this keeps working, we need to think about the storage and delivery method for clinical use. So you will have mitochondria The ideal thing will be to have autologous mitochondria. That way you will avoid any kind, you know, of rejection, draft rejection kind of thing. But there may be other methods for using kind of autologous mitochondria, or yeah, they're experts like MHC, so it should be like a kind of easier. So the storage, we have tried to storage mitochondria -80. They're pretty much stable. We were concerned about getting some crystals that may be like, you know, fracturing the mitochondria, but in the right media, they're pretty much stable. But of course, more optimization is kind of necessary for a long-term storage in case this actually works. So there are a lot of things that are still need to be done. So as I mentioned, we're trying different steps, transplantations like low calcium media for uptake, and then complete media for the maintenance of the cells. We're supplementing the cells for not, you know, giving them a chance to recover with some pyruvate, NDA, and uridine. We're also considering other kind of aspects of the mitochondrial transplant. And this is kind of very interesting because in physiology, in theory, mitochondrial transplantation occurs naturally. So if you have a cell that needs mitochondria, something happens, we don't know what happens, that will trigger a response to other cells. And one of the main mechanisms is that they will create like nanotubes. And these nanotubes, like a tunnel, basically, and they will be transporting proteins, and among them, they will be also transporting like organelles, like mitochondria. So MIRO1 is a very important protein for creating these nanotubes and for the mobility of the mitochondria. So we're actually experimenting if adding MIRO1 can enhance these mitochondrial transplantation experiments. And PEP1 is also a very important peptide, which will make mitochondria or whatever other thing that is coated with PEP1 to be more or kind of tastier to the cell, to be more suitable for getting like intaken into the cell. So maybe this way also, whatever extracellular mitochondria is in there, if they're coated with PEP1, basically it will be getting like integrated into the cell. So we have been thinking also in a kind of for storing this kind of mitochondria. So we have been working on a modified PRP, which we call the super PRP. It's basically PRP with another kind of extra additives, you know, like more glucose, more antioxidants, trehalose, or many other cryoprotectants are also actually being used in other kind of transplant, like organ transplants. So we're trying to create like a super PRP. And the route, this is kind of very important. So this is all experimentally. So we must consider like, okay, which route will be the best one? Intramuscular, intravenous, IV, or through the artery. So far, the few animal models we have been using, intramuscular is basically like one of the best ones. And we have seen like with the IV, a lot of rejection, basically of whatever is kind of injected. So where this is also like still work in progress. So there's a lot of things to do with the mitochondrial transplantation, and especially in JDM and DM. And a lot of things to uncover of the pathogenesis of these kind of diseases. So our main goal is basically to discover or unveil the mechanistic insight, the mechanistic part of why these diseases are causing these conditions in these patients. And okay, we know why, but we also know how to prevent or how to treat these diseases. So we expect that this project will be basically trying or help us in humanity to establish more biomarkers for these diseases, more targetable, therapeutic approaches, and more therapies of the mitochondrial transplantation. So, and if the mitochondrial transplantation works here, it will work like in many other different diseases. And just lastly, but not less important, there are many people involved in this project. The Lud Lab, which is my PI, the Johns Hopkins, Karolinska, rheumatology, and even NASA is involved in this because they want to try the mitochondrial transplantation in astronauts for avoiding aging and some muscle myopathies they have in outer space. So that's basically it. Thank you so much for your attention.
Thank you, Jorge, for this outstanding talk. It's a high level of science, this work that you have been doing this time. And also, I think that is a hope for these patients that we know that we don't have a lot of treatment options so far in these patients. For example, in the case of JAK inhibitors that you mentioned, you know that for EMA and for FDA are not approved so far, specifically for dermatomyositis or juvenile dermatomyositis. So I think that we have to do a lot, and this is of great value for potential the therapeutic strategies in these patients. So I don't know if the team have some questions or comments about it. Maybe we can start. I have some questions here, but maybe the team go ahead.
Thank you, Jorge, for your big presentation. It was great, really. And as I was listening to you, and I was thinking about my patients who have this dermatomyositis and those who have the calcinosis, they really get a painful disease, and they get really infectious diseases, and maybe they have distractions of the joint also. And as there are no many treatments, maybe we use the JAK-STAT inhibitions, but it is the last stage of the treatments you choose, because you have to try many different treatments and then you use the JAK-STAT. So I was wondering whether this study could just make us choose the first step, the JAK-STAT inhibitions, when you find these biomarkers, or when you could, I don't know, show that the interferon 1 type is upgrade. I don't know if you have read out something about that, or do you have something in mind?
Yeah, that's one of the points we're trying to do. So we're trying to, because usually these kind of treatments, when they have the calcinosis specifically, treatments are started like in a very late stage. So when the treatment is started, like sometimes there's nothing to do, or you can remove them like surgically, but they will appear again like somehow or eventually. And some of them are like almost impossible to get removed like surgically. So we're trying we are aware that using these kind of drugs sometimes is not like the best option if you consider like the immune response of the patient, because they can get also an exposure to any other pathogen, any other kind of infection. So it's also like, as you mentioned, like one of the last steps. So we're trying to make like more precise targets in these kinds of diseases, to try to use them like in an early stage without compromising the immune response of the patients, basically.
Okay, perfect. Any other questions? We have, do we have questions in the chat or not? Matthias, no? Okay. I have a question here about if you perform interferon signature in these patients. If not, do you think that is important, or maybe would be redundant?
It may be important. It may be important. So, I mean, we have found that this interferon signature is higher in the patients, specifically with calcinosis. But I think it depends in the comorbidities of the patients. But I think we will also need to consider, the balance of asking for different kind of studies on the patients, which are considered like the base studies of the patients, plus the other ones that may be still like undergoing some research, like these ones and any others. So I think we'll need to wait a bit more for having like a very precise response. Like, yeah, these patients or all the patients must be asked for this specific study. But so far, there's something there. So they have a higher like interferon response and signature, yeah. And to
Okay.
certain genes, not just like in general, yeah.
Okay. Okay. Also, other questions is about the cyclosporine. because you use cyclosporin H, right?
Yeah, not A. Yeah.
Cyclosporin A. And you know that cyclosporin H is not truly immunosuppressive as cyclosporin A. So why the election in this work, in this
Because
research?
that's a very good question. Actually, cyclosporin H, as far as I know, there's no like a lot of patients that have been gone into this kind of treatment. Okay, mostly it's like cyclosporin A, but the advantage of the H is that it specifically inhibits this specific receptor, formulated peptide receptor 1. So it doesn't do anything else to any other thing. So it just blocks this receptor on neutrophils. So by blocking these receptor neutrophils, neutrophils can still get activated by TLRs, for example, or any other innate immune receptor, but not through the formulated peptide receptors. So we're using that one because we're trying to see, okay, the formulated peptides from mitochondria, neutrophils, if we block their specific receptor, nothing happens. Or if we block the specific receptor, even by adding the plasma of the patients with a lot of FMET, the neutrophil still activates. Okay, there's something else activating the neutrophil. It's not just this. But what we saw is that the activation was mainly driven by this formulated peptide receptor, basically.
Interesting. Okay. Any comments?
Just an amazing work. And like when you get to this experimental point, it's always very nice to see like where it begins and how it's developing, and how great it will be. And it's already great, but the final result seems amazing. Like we are so excited to see your research.
Thank you for that.
Okay, and one more questions about the JAK inhibitors. That we didn't realize with precision about the JAK that you use. It wasn't one of the currently approved commercial molecules, or it was only for research?
I think we use only one. I think one of the current approved by FDA. Yeah, we have been
Okay.
using approved by FDA because the idea is to translate and like to move them to mouse and then humans. So yeah, that's something with that we need to think about with, for example, cyclosporine H. So yeah, we will need to develop probably like a certain drug or something that can, like an antibody or something that can target directly formulated peptide receptor 1 specifically. Yeah, because everything else we have been using is basically approved by FDA. Of course, except the part of mitochondrial depletion. I think you should not deplete mitochondria from humans, like just like that. So
Because, for example, we don't have in clinics the cyclosporin H.
yeah,
Only.
that's more experimentally. Yeah.
Ah, okay, perfect. And how long do you think that maybe this research will take to move to humans stage?
That's a very good question. So I mean, part of this work is done like other kind of colleagues that have been here like before me. Part is also made lastly, I mean, by me, and it's not just like a one-person project. So it's a whole team project, many disciplines, many specialties. So I think probably that if I'm right, hopefully we can see something happening with us, I mean, with our research or any other kind of researchers doing like something similar, maybe in the next couple of years or 3 years, because this is like a big boom, especially the mitochondria transplantation. Many people are kind of, not many people, but some important people are trying, and by important, I mean financially powerful people can do a lot of experiments. Some of them are also, of course, NASA. So when we got contact by them, they were like, "Yeah, we want to do this with astronauts." And I can very humble say I'm not a competition against NASA. I mean, if they want, they can do this like in 1 week. But the idea is to work together. So there's a lot of work behind this. So hopefully, like in the next couple of years or 3 years, we can see something, at least like clinical trials or something like that.
Okay, maybe because we can like save a lot of immunosuppressive drugs in these patients with this kind of treatments. And also about the origin of these mitochondria, do you think that maybe you can explore in allogenic mitochondria or only autologous for human? What do you think about it?
Yeah, I mean, the only thing is, I think it will end up depending how pure your mitochondria are at the moment of the isolation. Because in theory, mitochondria per se, they don't have like, it's not like a cell. They have many kind of common things with like a cell membrane, stuff like that, but they don't have like the MHC and stuff that can be like, causing like an immune reaction, like a draft kind of reject reaction. But the only caveat there is that mitochondria have some peptides, because they come from bacteria, that may be immunogenic. So if you have like very pure mitochondria, like perfectly shaped, very functional, they may work. I mean, allogenic, that will be like perfect. But if you're injecting mitochondria that somehow are kind of, broken, or they're kind of exposing some mitochondrial DNA or things like that, you may be careful, because you can get like the other effect. You may be causing like an immune response, like an inflammatory response through the mitochondrial DNA, or generating antibodies to mitochondrial DNA, like you can see, like, autoimmune diseases and stuff like that. So I guess it depends, like, in the purity of the mitochondria, how pure you can get the mitochondria. You can,
Yeah, especially because they are difficult to the manipulation, right, of the mitochondria is very fragile.
yeah. So there's some kits available to isolate mitochondria. It usually takes maybe like half an hour, but the way we do it, since we try to be extremely careful, we do it like manually, basically, and it takes basically like 6, 7 hours to get like maybe 20 million mitochondria. So it's like a whole day of mitochondrial isolation, basically. Yeah.
Okay, okay, perfect.
I have another question for you, Jorge. As you know, these kind of diseases, dermatomyositis, they have maybe some relation with some cancer or tumors that can
Yeah.
appear in some different moments of life. Maybe when you see adult patients that they don't get really good responses to standard therapies, maybe that's the risk factor to maybe develop this calcinosis, or maybe then cancer appears. In this way, there are some autoantibodies like NXP2 that maybe it is the more related to calcinosis. And then it's another one, which is the TIP1, which is also related to cancer. So did you consider these kind of related diseases?
Yeah, that's a good question, Matthias. That's a really good question. So we are subdividing like the groups, especially in DM, like MDA5 positive, T1 gamma, which are more associated with cancer. So we're also like considering the risks they have like with this subdivision, to see if there's somehow like an association also like cancer, calcinosis, so risk, or interferon mitochondria dysfunction, calcinosis, cancer. So we're trying to play there like by statistically also, like, to see any kind of connection. But that's a good question, yeah, and something to consider, absolutely.
Okay, I think that we don't have more comments or questions. So, Jorge, thank you very much. This is truly like the future of medicine that you presented your
Okay.
work. So, we are grateful with this work, grateful with your time. Thank you for joining to us. This is your home, of course. This is lastly, you are part of Latin America, your country is Mexico. So, of course, you always will be welcome in our
Thank you.
community. And we hope that this is not the first and the last time that we have with us, right? So thank you very much.
Thank you. Gracias. Obrigado. Thank you. It was an honor.